Okay. Let's see if anybody's. Oh, they're coming. I won't close it.
Aww.
What time is it? If it's 5:01, I can close it. If it's on 5, they're on
You
10.
closed it on me when it was 5 before.
No, if I closed it when it was at 5.
Yes.
It was at 5.
Oh, there, I got lucky. They got lucky then. There you go. All right, vector competence. Define for me vector competence.
How did vector explain
Good. Okay, so how well the like sometimes I try to like write too fast, and then my handwriting suffers. How well the vector spreads the disease. Okay. So I want to talk about like all the things that can potentially go into how well a vector might spread a disease, and all the things that can change to cause a vector to have changes in their vector competence. Okay. Let's think about first within the vector. What are some things that you can think of that, let's make a list, and let's see if you get some of the things that I thought of. Things within the vector that can cause changes in vector competence.
Moving on to different life stages.
Oh, so you mean like be like transstadial transmission of like a pathogen? So you're saying life stage transitions. I did not think of that one.
Are you talking about like components that would contribute to competence, or like?
I'm thinking of like sort of, my perspective is things that you can think of that will clearly like have an impact on this process. I suppose it's pretty expansive.
Like how many blood meals, like
how.
Yeah.
Yeah.
What? Okay. So feeding. Yeah. Feeding.
like how we call this in the first classes when we were discussing filariasis and how the filaria came out only at night.
Periodicity.
Like
Periodicity.
like if there's
So-
an overlap between the host availability and the vector presence.
Like overlap.
Yeah.
Yeah. Okay. I'm realizing this is going to get like extremely expansive. Let me go through my list, and then we'll add things that I did not think of. Okay, so insect, innate immunity. What is innate immunity?
Like natural resistance to it.
Yes, but more specifically, like there's innate and then there's adaptive immunity, right? So what's the difference between innate and adaptive immunity?
Innate would be like genetic.
Yeah, it's like at the cellular level. So like an insects don't really have time for adaptive immunity like we do, because insects live essentially some insects live long.
So the white cells of the insects are chemocytes?
They have hemocytes, yes, that's actually on my list. But it's not adaptive in a sense of like there's pathways that will activate hemocytes to do certain things, but it's my understanding that they're not making like antibodies.
But
and stuff
they
like
don't
that.
have like hypersomatic mutation or light chains and light chains rearrangements.
Well, they definitely have like rearrangements of stuff on the outside.
But I mean, in terms of like the B cell and the T cell response, when they encounter an antigen and they go through a rearrangement of the light chains from the immunoglobulins to adapt that, like they don't have all these.
Here's what they don't have. Here's what I can definitively say that they don't have. Like if a human gets infected with a virus and you have this whole immune response and then you get over the virus, you can become completely immune. Insects not like that. Like insects will either get the disease, and then essentially their lifespans are not long enough. But okay, with respect to kind of what you're talking about, like let me talk about some of those systems. Okay, so they have antimicrobial peptides, AMPs, which are typically things that will so if a pathogen enters an insect, you're right, there will be like an immune response from the hemocytes. I'll put a pin in that in a sec as I get down to the other pathways. But one of the factors that they can create that will start attacking the pathogens are antimicrobial peptides. Okay, and they typically insert into membranes. They have like a typical like mode of function. So if you have like a pathogen, whatever, this is the pathogen, these antimicrobial peptides will insert into the outer membrane of the pathogen, and then typically they just like form a hole and it kind of like pops the cell. And that's typically how they're like killing cells. Okay.
Question for like basic understanding. For most of these pathogens, are they gonna mount an immune response against it, or does the pathogen have an effect, negative effect, stimulate a response from the insect, or it just exists there?
No, this is definitely a stimulated thing. Okay, and there's two pathways that go into that. Like they were my C and my D, but we might as well talk. Oh, actually, okay. So let me answer that 1 second. B is hemocytes, which is brought up. So insects do have the circulatory system, and they do have something that are like white blood cells, which will phagocytose pathogens, which are the hemocytes. Okay, and within that, there's cellular pathways, the toll pathway, and the IMD pathway. So these are essentially like, there are receptors, so there are toll receptors and there are IMD receptors that are on the outside of the hemocytes, okay? And when certain pathogens enter the insect, certain ones of these receptors will recognize them. So the IMD pathway is responsible for recognizing gram-negative bacteria, for example. And so what would you think that the IMD pathway potential receptors would be like binding onto?
OMPs?
What? Well, probably OMP antigens, but also the peptidoglycan in the cell wall of the gram-negative bacteria.
So all of these are receptors that recognize pathogen-associated molecular patterns, right?
These are pathways, and within those pathways, they have receptors on the surface of the hemocytes that will recognize certain pathogens. And then when they recognize it, they'll turn certain things on or off. So for example, if the IMD gets recognized, it will turn on, or it gets activated, it will turn on the antimicrobial peptide expression. And then they'll start like pumping out antimicrobial peptides into the hemolymph. Does that make sense? This one leads to, the toll pathway leads to phagocytosis of the pathogens. So right, some hemocytes are gonna be actually capable of inducing receptor-mediated phagocytosis. They'll suck up the pathogen, and then depending on what it is, there's different pathways to destroy it, but destroy it internally inside the cell.
It's like a macrophage?
Yeah, it is like a macrophage, yeah. Okay, so essentially, the point is, okay, wait, there's one more. A, B, C, D, E. There's one other like big one, which involves suppression of RNA-based viruses. What would we think that is? Virus people should know.
There's only one.
You're what?
Can we repeat the question?
Well, there's an innate immune response in insects that can suppress viruses, RNA viruses.
Like silencing genes?
Yeah, well, kind of. Well, I wouldn't call them like silencing genes, but there's an RNA.
Yes.
There's RNAi systems.
Yeah.
What? You say microRNAs? That's another. That falls into the RNAi system.
Yes, RNA.
Yeah. So So there's a few different RNAi systems, like if we want to quickly go over them.
Is it the I is for interference, right?
That's correct. So it's RNA interference. I can explain that quick. I don't have much room. I'm thinking of what to erase. I can erase this. I'll just start erasing from the top down. So I won't expect you to know too much of the molecular details of RNAi, but there's a few different systems. I'll just lay out the two main ones. There's the siRNA pathway, and then there's the microRNA pathway. Okay, and what happens in RNAi and RNA interference? First of all, what is RNA interference? What happens? So RNA interference is a system where if it encounters some type of message or transcript, it can shut it down or turn it, like lower the levels of it through a process of nuclease degradation of that signal. Okay, and so these are essentially pathways that turn on that will cause destruction of RNA, certain targeted RNAs, which potentially is a virus. Okay, so the way that they're stimulated is the cell in some way the cell always sees double-stranded RNA. So you get some bolus of like double-stranded RNA. In this case, in small interfering, it's actually double-stranded RNA. So it's actually like one strand and then another strand that are hybridizing to each other. And so my virus people, what viruses maybe would show them this?
Excuse me?
What viruses might show them this type of molecule? So flaviviruses are going to be something like this potentially, but a bunyavirus, right, can be the ambisense. So it could produce a molecule that looks like this.
Sure.
We have RNA binding to each other. Does that make sense?
Yes, sir.
Okay. And when the cell sees this, is this normal or abnormal in a cell for an insect?
Abnormal?
It's pretty abnormal. This is not something you normally see as an insect cell. So when it sees this, there's a system that recognizes it, which is these RNAi systems, the siRNA. There's a protein called, now it's going to get really complex. There's a protein, we'll just do it as stepwise what happens. There's a protein called dicer. Dicer sees this and it cuts it up into little pieces, okay? And then there is a protein called argonaut, and that's inside of something that's called the RISC complex, okay? And what this then does is this grabs these little chunks. So dicer is just like it sounds like a cooking show, like it's dicing them up, cutting them up into little chunks. And then this protein called argonaut grabs onto them and essentially it cleaves the other side of it. So now if you have one of these little chunks that you've cleaved to the other side of it, and it's bound by a protein, which is part of this RISC complex, this is now essentially like a homing beacon, this is a nuclease, and it's a homing beacon, and it's going to recognize anything that binds to or matches this sequence. Does that make sense? And what does the sequence come from?
Those?
No.
Or the pathogen?
The pathogen. So now it's going to be a targeted nuclease that targets the destruction of a potentially like a viral pathogen. Does that make sense? Okay.
So the dicer is seeing them at the nuclei level, and then they're in the cytoplasm, they mark these exogenous genome parts to be marked for the RISC complex to eliminate them, right?
Yes, so Dicer's process is when it sees the initial double-stranded RNA, it cuts it into little chunks. And these are then little chunks that are of potentially a virus. And then the signal can be amplified because Argonaut grabs some of these chunks, or all these chunks like individually, and then it removes one piece of it. And so now this is a homing targeting mechanism, because this is a sequence, right? This is A's, if it's RNA, A's, U's, G's, and C's, okay? And these are exposed now looking for hydrogen bonds, so they're looking to find like a partner, these hydrogen bonds, and they're only gonna bind with something that they match. So essentially then you have now this nuclease that's bound to this thing that's looking for a partner, and the only thing it's gonna match with is another sequence from the virus. Then once it finds it, the RISC complex cuts it up and destroys it, essentially. So if there's another molecule, like a virus, part of the virus genome that matches, it'll grab it, and then it will cut this transcript. And that's how, essentially that's how RNAi works. Does that kind of make sense? Or should I go again? Should I go again? Should I slow down? What do you think? Good? Okay. This pathway is exactly the same thing, except the only thing that's different is the bolus of RNA is different. In the microRNA pathway, a microRNA is a hairpin. So it's a single RNA that hairpins and binds to itself. But this still creates a weird double-stranded RNA molecule, okay? So that's the only difference between the small interfering RNA pathway and the microRNA interfering pathway. Everything else is the same. Dicer sees this, grabs it, cuts it up into the chunks, and then Argonaut grabs these chunks and makes them essentially single-stranded. And Argonaut is part of this bigger machine, the RISC complex, which now is holding on to these things. And then they seek out their target, and they'll destroy then RNA viruses. Okay, so that was a lot of kind of molecular biology detail. What I would expect you to know on the test is just what is RNAi? What does it do? It's a system that degrades messenger RNA or viral RNA, and it's an innate immunity system. So this is sort of like a list of some of the innate immunity systems in insects, okay? That's what I expect you to know about it. And the whole point of all this, is how can this affect vector competence? It's pretty easy. These are the ways that the insect has to destroy pathogens. And so if these pathways are particularly good at destroying a particular pathogen in one vector, then it would have low vector competence. These are also pathways that can evolve and change insect to insect. And so again, if you're thinking about why one insect has high vector competence or low vector competence, it can play into these immunity systems. Maybe they have better or worse immunity systems for some particular pathogen. Does that make sense? Okay, so that was a lot of just sort of one of these mechanisms that can change vector competence, insect immunity. Gut conditions. How can gut conditions increase or decrease potentially vector competence?
pH.
Mm-hmm. How would that affect things?
Oh, I don't know. I'm assuming it has something to polarity, maybe.
Yeah, it's going to change protein interactions. So imagine you're a pathogen in the gut of some insect. Right, one of the first things that needs to happen is it's got to go through most pathogens have to get out of the gut and go through the basal lamina. And
How is that the physiologic condition in mosquitoes called? The mid-gut barrier or something?
Say that again, what's what called?
How do they call this condition of the mid-gut barrier preventing the viruses infect the cells? How do they call these?
Well, there's in the gut, if you were to expand the gut, there's a wall of the cells.
Yeah.
That's typically called the. I'm trying to think.
Yeah, when the mosquito is not permissive to the virus.
What do you call that?
Because there is a barrier in the mid-gut, including the cells and the peptides that they produce. How do they call this?
What would they call that? I think you just say it was low vector competence. What you're saying, is there a specific term that specifically defines?
It sounds like the proventriculus, the way it was.
Well, that's a physical structure. The proventriculus is a structure in the gut. He's wondering, is there like a term for if a virus cannot get out of the gut?
Oh.
I don't think there's a specific term for that. What I'm trying to say is that would be, you'd call that low vector competence. Unless there maybe is a specific term. But I've never heard it. Okay, so anyway, like the point is, yes, gut conditions, pH, some insects have basic, some insects have acidic guts. It's like different, insect to insect. And that's gonna play into the pathogen, the proteins on the pathogen surface, and whether or not those proteins then can have their proper interactions. What else is in the gut that can cause potential issues?
Microbiome.
Yeah, that was my number 3. So the symbionts. But before we go to the symbionts microbiome, what else are guts famous for?
Digestive enzymes?
Yeah, there's going to be, like that's what the gut is full of, is these enzymes, they're proteases. So there's gut proteases. And like the activity of these gut proteases, if they are impacting the pathogen, that's going to in a negative way, that's going to lower the vector competence. Symbionts microbiome, yes. So we talked about the Wigglesworthia, Sodalis, our gut symbionts in tsetse flies. Mosquitoes often have a bacteria called Serratia in their guts. There's a bunch of these. Like you don't need to know them specifically. Just know the idea that like symbionts in the microbiome of the gut of the insect is a known factor that will raise or lower vector competence. And then for the gut receptor and like evolution of those. So again, in most situations, you have a pathogen in the gut. It's got to get through the basal lamina. The first part in that process is it has to bind and recognize those cells that are on that lining. And there's always going to be a receptor-mediated interaction where the pathogen has some outer surface protein that's grabbing the receptor. So if there was a SNP, single nucleotide polymorphism, or some like mutation in that gut receptor, that can lower or enhance the vector competence. If it makes the interaction stronger, it would increase the vector competence. If it makes it less strong, it would decrease the vector competence. Okay. Once it gets out, let's see. Okay, so there is a specific term for this. The ability to sort of like, it's kind of a weird thing I wrote. The ability to find the salivary glands. So there is actually a specific term for this. It's called the maturation index. Maturation index. The maturation index is defined as a percent of which, the percent of which, say you have a tsetse fly that has taken up trypanosomes. What is say you have a population, say you have 100 tsetse flies that have sucked up blood meals that have trypanosomes. What percentage of those infections get to the salivary glands? So the percent that get to salivary glands, whatever that percent is, that's the maturation index. And there's actually some like interesting, so you can imagine scenarios. Let's expound on this. You can imagine scenarios now where you start to like calculate values of vector competence. And that's a thing within the research is people are always trying to quantify, is this vector better than this vector? And you have to imagine that there's this like chain of events, right? Like if this chain was the probability or the percent that pick up gut trypanosomes. And then maybe this is the percent that break the gut. And then maybe this is the percent that find the salivary glands, the maturation index. And you can imagine now that vector competence is going to be actually kind of like a mathematical calculation of this probability times this probability times this probability times this probability, etc. And for each host, like there's going to be different values for all this. So for example, like this is just a simple calculation, but for Glossinia morsitans, okay, the infection rate of trypanosomes is about 10% trypanosomes, okay, and for Glossinia, a different species, Pallidipes, the percentage is just something about 1%. Okay. But, and so you might say Glossinia morsitans is obviously a better vector. Okay. But that's not necessarily true because this is just the first point in that chain. Does that make sense? Like you have to think through the whole process. And so if you actually start then calculating in the maturation index of Glossinia morsitans is 20%, okay? And the maturation index of Glossinia pallidipes is 90%. So this one, if it gets the trypanosomes in the gut, they're much more likely to get to the salivary glands. Whereas this one's a reciprocal. It's less likely to, or it's more likely to pick them up in the gut, but they're much less likely to get to the salivary glands. So then if you just multiply these, this would be 0.1 times 0.2. That's essentially like 2%. 2% of the tsetse flies become sort of like transmittable. If you're only including these like two factors in that sort of like calculation. And this one is 0.1, or it's 0.01 times 0.9, which is about 1%. So then again, like if this is just a simple calculation, but then you conclude like, okay, this one is less vector competent than this one. But do you see, like the main point of me showing you this is like the breaks in the chain, the series pieces in the chain can be completely different, can be totally different numbers. And you don't really have a good idea of the vector competence value unless you've thought through like the whole chain process. That's the point.
That seems like a very complex algorithm.
No, it's just the
No,
probability.
not this, but
Oh.
in actuality.
Oh, like how to actually calculate it. Yeah,
Yeah.
it is. Like, oh, so, and that's actually like worth talking about. Like what, there's a lot of things that go into this, and there are sort of like different values that people put for that. One of the things, there's a formal equation in medical entomology, which is like how you calculate, not quote unquote the vector competence, but the vector capacitance. Like there's a bunch of variables for this, but some of the things that go into that equation are like bite rate. So some mosquitoes, like they're more aggressive, like, and they'll bite humans more. And so you can imagine going into this equation would be bite rate, and then the percent of bites that spit out the pathogen. Like, yes, it gets very complicated. So yes, I fully agree with your point. Okay. I did have the feeding behavior as we were doing that list of a whole bunch of things in the vector that can contribute. The last thing I had was the sex, male, female, and into that went the biting, which Daniel said, the feeding habits, and then the final thing is within sex, male versus female, there's a whole separate chromosome, right? And there can be sex-linked genes that contribute to vector competence. So you can have male or female tsetse flies, and the vector competence might be completely different if it's male versus female. Not necessarily just the species, but also within the species, the sex can be a factor. Okay. Got it. Now let's think, so these are just, again, obviously I have not synthesized everything that can change vector competence, but I wanted to give you a list of some things that you can envision that can change or evolve that will change that value. Okay, let's think about within the pathogen. So evolution can, a vector can have a certain value, but the values that determine that vector competence value are not necessarily just things within that vector. They can be properties or genes present within that pathogen. So certain pathogens might evolve to transmit very well in some particular vector. So some things that can contribute to this would be like motile activity. So the ability of the creature or whatever to like move, to swim. That's oftentimes a part of this. Like I guess in rickettsia, the example was that RIC-A gene. That was one specific gene that we talked about that mutated, that contributed to its ability to essentially like move around. Let's see. I have a very specific, I'm trying to understand what I wrote here. We'll go to that later. What other things, like let's just make a list quick. What other things could change within a pathogen?
Like shape of the active site, or
So if it changed in the, so if the receptor site changed in the vector, maybe you'd call that some kind of like target site resistance or something like that.
Yeah.
But the thing that binds or grabs on in the pathogen can also change. So the receptors. And receptor evolution is like a big, like that is always a big area of research in medical veterinary entomology, is if you have this pathogen, what is the particular receptor that binds to whatever other particular receptor? And it's actually worth, like, there's a lot of papers on that. How would you scientifically like find the receptor? What would you actually like do?
Upstream, downstream stuff?
What do you mean upstream, downstream? Like experimentally, like what would you do to discover what's the key receptor? So maybe we'll do like a plasmodium example. So like if you have a mosquito and it's got a gut and you have a plasmodium in the gut, what experimentally could you do to figure out what is this interaction?
Like a yeast 2 hybrid.
How would you set that up though? So now we're getting graduate level questions, that's good. So in theory, like you might be able to use a yeast 2 hybrid, which is, a yeast 2 hybrid is a methodology that you can determine whether two things interact with each other.
Do you have like just all the interactors together on the plate?
Well, the problem with that, so the problem with doing like a yeast, I'm like, I don't know if I want to define how yeast 2
You can just
hybrid.
move on, it's okay, I was wrong.
Well, no, that's not wrong. Like you could make that work.
I mean, but for that, don't you basically have to like have an array of candidates and then
Yeah.
just throw stuff and see what sticks, basically?
Yeah, so that's actually what I was going to say was like the one reason why you would potentially not want to do that is you'd have to have, so maybe you would have like your outer membrane protein as you need a bait and a prey, essentially. Like you need bait and you need like a prey library. And so if your bait, you probably have a bait candidate, which would be like an outer membrane protein of your pathogen. So like maybe you're really interested in like OMP1, whatever, from whatever pathogen. The problem with the yeast 2 hybrid is you'd have to have an entire library that you screen it against, which is essentially like expressing all the potential receptors in the gut. But that might actually be like a good way to do it. Like it might be. The way that you'd construct that library is you'd have to, this falls into the RNA lab, you would essentially extract, like if it were me, if I was gonna have to do that, I would dissect out insect guts. I would purify messenger RNA from insect guts, from insect gut cells, and then you'd have a sample of all the messenger RNAs being expressed in guts. You would clone those into yeast expression libraries, and then that would be your like prey library. And then whatever hits, so again, like you don't need to understand the yeast to hybrid for this class, but you would do this experiment where you'd mix the two together, and then whatever hits you got would potentially be a first source of the interaction site. That's definitely like, that's a good way to start from zero, if you had no knowledge. So let's say you develop your candidates from doing that. Let's say you did that. How would you prove that let's say you get a series of like 10 candidates that were your hits from the yeast to hybrid. How would you then prove which one is real? What would the experimental pipeline look like after you had identified some of these candidates?
Oh God, I don't know, would you have to like silence it in the bug and see if it could get infected?
Yeah, that's a really good idea. So in some cases you could use RNAi. You could use RNAi can also be used as a methodology. You could knock down these candidates individually, and then what would you measure? Hang on, what were you gonna say? So let's say you did do RNAi, or so RNAi would do a knockdown. So you essentially like knock down whatever these candidates are in the gut, or you could do a knockout. Like if you could make a transgenic mosquito and you say you made 10 transgenic mosquitoes, all knockouts, then what would you look for?
No infection, or like lower titer?
You just essentially do like microscopy, right? Like you could knock down these candidates in 10 different mosquitoes, and then you would just do microscopy and see if they were the correct interaction, the pathogen would never get out of the gut. But if you did microscopy and you saw the pathogen in the hemolymph, then like, what do I want to say? Do you understand what I'm saying? Like your readout would be simple microscopy of where is the pathogen. And you would want to find the candidate that when you knocked it out, they never get out of the gut. That's what you want to find. And that's what they do for like finding these interactions. That's my point, is they first somehow through scientific methods establish like a list of candidates. And then after they have a list of candidates, they'll do a more thorough like knockdown or knockout, and then they'll just do microscopy to see if the pathogen stays in the gut. And if it stays in the gut, then you found the right thing. And then potentially that's an interaction then that you can like exploit. Okay, that was kind of like, I'm doing my best. But this is like if we're trying to think of like experimental pipelines of how you actually find these key interactions that are key for vector competence, that's something that you can do. Okay, let's see. You can also have factors that vary vector competence outside of the pathogen and outside of the vector. So like these would be like extrinsic, totally separate from extrinsic incubation period, but these would be like extrinsic factors that contribute to vector competence. Let's make a list of these. What would go into that?
Temperature and humidity.
Yes, that was my first thing, temperature. And then also like wintering. Winter is super important for insects. So insects are super attuned to temperature. Pathogens are super attuned to temperature. And then when winter happens is key, and the conditions of winter is also key for things like ticks, where they might pick something up in one stage and then they actually have to go through a whole nother stage and overwinter before they bite again. What else? Daniel said the geographical overlap. So that's the case of like if you have two different tsetse fly species, but one is always in the deep jungle, that one is never going to have high vector competence because it's literally never going to be overlapping with humans. They have to overlap with both the potential source that's getting the pathogen and the reservoir. So there might be one particular tsetse like Glistinia morsitans, which overlaps with the mammals that it's eating, picking up the pathogen from, and the humans. What else? This is kind of easy, I guess. What other factors? We'll put seasons. What else can happen? What do animals do that they're famous for, like moving around? What's that called?
Migration.
Yes, so like migrations. Maybe maybe something that was not high vector competence all of a sudden changes and becomes high vector competence because all of a sudden some crows that have West Nile virus have migrated to that spot. That can happen. So there can be quick changes depending on like if reservoirs are moving or migrating. That can cause changes in vector competence. One thing I'm thinking of that, think about like, I'm trying to, how would I hint at it? Trying to think of like a hint. What do we do? Like we have behaviors. Humans have behaviors.
Like urbanization.
Yeah, so for sure. Okay, so let me broadly say human behavior. And a lot that plays into that is urbanization. That's a good one. So in the rainforest, one of the concerns is as humans start cutting down the rainforest and get closer and closer essentially to these like weird zoonotic cycles of the pathogens, you create situations where now you're exposing new humans to these new pathogens. That's definitely one. Migration of humans would go into that. Also just to what would you call it? Somebody said diurnal patterns. Humans have diurnal patterns too. Like in some situations, the first thing you might do when you're waking up as a human, you might go get some water. You might go to like the water hole. And if the vector is programmed to be out biting at that time, that can then create a situation where it has high vector competence. Okay. Now here's an interesting thing. How much time do I have left?
8 minutes.
Okay, good. Okay, this is perfect. Okay. So I actually want to answer a question that you had, which was, I think you asked about the distribution of brucei, which is the nagana, but also too, I think you asked the question of, do humans get brucei? And so I found the answer to that, and it falls within sort of like a discussion of competitive evolution, of like host pathogen evolution. So there is a protein in humans, a gene called TLF. That TLF stands for trypanosome lytic factor. Okay? And this is a lipoprotein. What's a lipoprotein? Yeah, so it's a protein that it's got fats on it, and typically these can insert like into the membranes. So always talking about like obsessively talking about like things on the surface. This is a special protein that if it's phagocytosed by trypanosomes, it'll kill the trypanosome. So humans actually have a gene that specifically like is a resistance gene for us against trypanosomes. And the one that it will kill is Trypanosoma brucei. So this protein is lethal to Trypanosoma brucei, and that's the reason why humans are not getting the brucei disease. That's why we're not getting the nagana. It's essentially in primates, this gene. So that's a case where primates have evolved a specific resistance thing that gives them resistance against this. And one of the reasons so in the others, this is super interesting. This is where the competitive evolution comes in. In the other species, the gambiense, or not species, but strains, Trypanosoma brucei gambiense and rhodesiense, I can say it now today. In these ones, they have a resistance gene to the TLF, which is called SRA. So this is a separate gene. Genes always have like weird like annotated names. SRA stands for serum resistance associated. So probably how this got named is they were screening for genes that gave resistance, and they found some gene and they gave serum resistance associated gene, and so they named it SRA. Okay, so SRA is a gene that these two strains have, but these one doesn't, that when the trypanosome encounters the TLF, it negates the effect of the TLF. And so that's why these ones do infect humans, and our immune systems cannot, or our protein cannot kill them, is because there's literally like this competitive evolution happening. And so you might, like last time I talked about, one of the big concerns in trypanosomes is because they're eukaryotic, they may mate. One of the big concerns then is if there's ever a situation where these ones are somehow mating, brucei might somehow pick up this gene, perhaps, and then that would make brucei able to infect humans. Like that would be a big problem if that happened. So here's just an example, one specific example where I actually found like specific papers that define specific genes that are competitively evolving and causing humans to essentially either be able to be infected or not infected because of a specific gene that they have. Any questions? Good? Okay, that's all I got. How much time do I have left? Is that about it?
3 minutes.
Okay, good, that was good. Okay, so that's vector competence, that's my spiel. What's the next lecture I have to look? I have to look it up, but I'll post it in the Discord. Have a good weekend.